Hello guys, welcome back. This is Dr. Hadi here. Today's topic is, of course, allosteric regulation. But before we start allosteric regulation, I would like to know the word regulation of enzyme. And then we will come back to the allosteric regulation. Dear students, as you know that inside our body there are thousands of chemical reactions are taking place, and all of these chemical reactions involve the conversion of reactants into product. In a very simple way, these chemical reactions are continuously taking place inside the body in order to survive the body, in order to establish the basic functions of the body. Of course, without this enzymatic chemical reactions, no life would be possible. Okay, so now what I am going to share with you, that is what is mean by regulation. Many students, they even don't know the word regulation. That is why they face lot of problems in allosteric regulation. So regulation, in order to understand regulation, let me tell you something, that suppose this is the reactant A and it is going to convert into the product B. Of course, this reaction is facilitated by the enzyme E. Suppose that the product is very less or absent in the body. Now when the product is desired, product is necessary for the body, but at the time this product is absent or less, what the body will need now? Of course, the body will need more of B. The body will need more of product B. This time body needs increased product. And another situation, inside your body there is reactant and the product, but this time the product is in excess, more than required. So in this situation, what your body will need when the product are too much, then our body need to stop the product B. Body needs to stop the product. Body needs to decrease the product now. So I will bring this arrow downside. This was upside and this was downside. This up and down in our body is called regulation. This is some kind of control inside our body. When something is in the normal range, and sometimes it happens that things go up from the normal range, then we need to bring them back to the normal by decreasing it. And sometimes things go down from the normal and we need to increase it in order to come to the normal. This situation is happening inside our body. Means a lot of chemical reactions need to be controlled. A lot of chemical reaction need to be regulated in terms of this ups and down. So the definition of regulation is the control ups and down. The control of the enzyme activity in the body is called regulation. You cannot leave these activities inside your body as such, because it will harm your body. If the product is required and your body does not have this product, this will harm your body. If the product is in excess, this will again harm your body. This is dangerous to your body. So your body need to decrease this. So at one place we want to increase the product, in other place we want to decrease the product. This increase or decrease of the product is called regulation. Finally, we got regulation. Now there are several ways inside our body where the enzyme activities are controlled by different means, by different ways. There are more than one ways. So in today's lecture, you will be taught only one way. Of course, the remaining ways will be discussed in the upcoming lectures. So what is the first way by which the chemical reactions of the body are regulated? The first way is the allosteric regulation. You got the background. Now you come to the main topic of allosteric regulation. So if you are a student and you are at the examination hall, the very first line that you would like to write about allosteric regulation, it will be that allosteric regulation is a part of regulation of enzyme. This is the first statement. Okay. Now, in order to understand this heading, I would like you to come to this diagram where we have an enzyme. This is just a simple diagram of an enzyme where you can see there are two sites. Site number 1 is this one that we already know that this is called the active site, and at active site usually another chemical molecules attached, right? And these chemical molecules are called what? Substrate. I don't want to repeat this again. So this active site is for the attachment of the substrate. This is for the attachment of the substrate. Beside this active site, there is another site on the same enzyme called the allosteric site. This is our allosteric site. So now we came to know that on the enzyme there are two sites. One is called active site for the attachment of the substrate. You know very well what is substrate. And another site is for the allosteric site. And we will discuss that what is the purpose of this allosteric site. Okay, so just like active site is for substrate, in the similar way, the allosteric site is for the attachment of this one, allosteric effectors or modifiers or modulators. So here at the allosteric site, some other chemical substance will attach, will bind. These chemical substance may be called modulators or modifiers or allosteric effectors. This is the first point. Now, these effectors or modifiers or modulators are of two types. Number 1 is the positive allosteric effectors. The first one is called positive allosteric effectors. Second one is the negative allosteric effectors. Positive allosteric effectors are those molecules which activate the enzyme after they bind at the allosteric site here. Once these molecules bind at the allosteric site, the enzyme will start functioning. The enzyme will start functioning. Of course, the enzyme will act on the substrate S. The enzyme cannot act on the substrate S until and unless this positive allosteric effector attaches here at the allosteric site. So this is a kind of switch button. If this button is on, the enzyme active site will functioning. If this switch button is off, the enzyme active site will not be able to attach the substrate and perform the reaction. So such molecules which activate enzyme after binding at the allosteric site, they are called positive allosteric effectors. Okay. And for these molecule, the allosteric site will be called as activator site. Bind at the activator site. So my dear students, of course this is called the allosteric site. But if at this site the positive allosteric effector is attached, then this site has got another name, second name, that is called activator site. So you will call this site as a activator site. Now we got two names for this site. It is of course allosteric site, but after activation we will call it the activator site, because positive allosteric effector will be known by its activator site. Now the negative allosteric effectors, these are also small molecules. If these molecules attached at the enzyme's allosteric site, as a result of their attachment, the enzyme fails to carry out its action, the enzyme activity is completely blocked, enzyme cannot attach substrate, then we will call these molecules as a negative allosteric effectors. Negative allosteric effectors are molecules. Don't worry, these are molecules, but their attachment at the allosteric site cause the inhibition of the enzyme. Therefore, we add the word negative to these molecules because they are stopping your enzyme. They don't allow your enzyme to start reaction. Therefore, they are called negative allosteric effectors. Now, that attachment site will be called another name, just like in the previous case we called the allosteric site as the activator site, but in this case we will call the inhibitor site. That we will call it the inhibitor site. Inhibitor site. Okay. So these were two types of molecules that are involved in the allosteric regulation. Now I will tell you the mechanism of allosteric inhibition, the mechanism, that by which mechanism this happens. This is really very important to know. Okay, if you want to learn the mechanism, exact mechanism of this allosteric regulation, you need to focus on the enzyme, the three-dimensional shape of the enzyme. As you know from the FSC level, from the intermediate level, that an enzyme consists of, this is an enzyme, let's suppose this is an enzyme. The enzyme consists of small units, small molecules. Okay, small molecules. You know very well what is the name of these small molecules. They are called, of course, amino acids. So the allosteric enzyme, what are allosteric enzyme? Those enzyme which contain allosteric site, they are called allosteric enzyme. And you need to know, you should know that allosteric site may not be necessarily present on all the enzyme. Allosteric sites are present only on some specific enzyme. Fine. So definitely they will be called allosteric enzyme. at the surface of the allosteric enzyme, lot of amino acids are arranged in the form of flexible protein. Flexible, what do you mean by flexible? Something that can change its shape from one form to another form. Let's suppose this is one form of the enzyme, and after it changes its shape, it become like this. So this flexibility of protein results two forms of enzyme. There are two shape of the enzyme. That is the relaxed state and the tense state. An enzyme, especially the allosteric enzyme, keep itself in two forms. One is called the T form, another is called the R form. T stand for tense and R stand for relax. These two are the different shape of the same enzyme. And interesting thing is that one enzyme can have both the T shape as well as the R shape. I mean, you can see that one enzyme, let's suppose this is one enzyme. This is one enzyme. 50% of this enzyme will be in the T state and 50% will be in the R state. Both relaxed and tense state will be there. Important thing is now that what or who will determine that enzyme will be in which form? 100%. Who will determine that all of the 100% of the enzyme will become either in the R form, relaxed form, or total enzyme 100% will become in the T? Who will determine that enzyme will become in completely relaxed form or in the tense form? So that is again the effectors here that you saw, the effectors, the modifiers, the modulators. The modulator will determine how. In case of your positive allosteric effectors, your positive allosteric effectors, they will bind at the enzyme. Now see here, this is again the enzyme. We know very well that at the enzyme, some of the part of the enzyme is in the tense form and other is in the relaxed form. And interestingly, the T form can be converted into R form and the relaxed form can be converted into the tense form. So both are in equilibrium. Okay, we just need one push. We need one thing. We need one person to come and tell enzyme, hello Mr. Enzyme, please convert your whole body from the tense into the relaxed form or from the relaxed into the tense form. So who will determine? The positive allosteric effector, which are small molecules. These molecules, when one of these molecules combine at the enzyme allosteric site, what will happen? This enzyme will completely convert into the relaxed form. Now this is the activated enzyme. It is 100% R form. There is no T form now. Therefore, this is the active enzyme. This is what enzyme? This is now our active enzyme. Because positive allosteric effector favor like the R form. They like that. What I have mentioned, allosteric activators, you can call it positive effectors. Allosteric activators or substrate favor. Favor means they like. They like which form? Relaxed state. Whenever they attach with your enzyme will shift into the relaxed state, R, which is the active state, which is like this. Now a reaction could take place. And on the other side, if another molecule which is called negative allosteric effector, if this allosteric effector is attached at your enzyme, what will happen? Complete enzyme will be now in the form of T form, that is tense form. This tense form is what? Is the inactive form. This is what? The inactive form. So now 100% of your whole enzyme is in what form? Is in T form. So in this way, inside your body, an enzyme can be activated when required. An enzyme can be deactivated when required. So both activation as well as inactivation is there. Activation, more product. Inactivation, less product is there. This up and down is called the regulation of the enzyme. So you saw one of the way by which the enzymatic activities are regulated, that is called the allosteric regulation. And you saw that how allosteric enzyme are involved for the allosteric regulation. Why this topic is called allosteric regulation? Because the enzyme which are involved in this regulation, they are actually allosteric enzyme. And why they are called allosteric enzyme? Because they contain allosteric sites. And what are allosteric sites? Allosteric sites are those sites which are besides the active sites. Which are besides the active site, means active sites attach substrate, the allosteric site attach the effectors. And one important thing is that, which very much important thing is that this allosteric site influences the shape of the active site, means the active site shape could be changed from this form to this form. That is all under the control of the allosteric site. Means allosteric site will determine whether the active site will attach the substrate or not. This is totally under the control of the allosteric site. If now allosteric sites are under the control of molecules, if a molecule, let's suppose this is one molecule. If a molecule attaches at this allosteric site, as a result of this attachment, the active site binds substrate S and convert into product. We will call this molecule positive allosteric effector, and this site will be called as activator site. That's what it will be called, what? Activator site. On the other hand, if we have another molecule B and it attaches at the allosteric site, as a result of this attachment, the shape of the active site changes like this. The shape of the active site changes like this, means a kind of a tense form. As a result of this, the substrate cannot attach. When the substrate cannot attach, the substrate cannot be converted into product. So no product, no reaction. As a result, we will call this molecule as a negative allosteric inhibitor. So this was the whole video lecture about the allosteric regulation. One example is also there. A student might be thinking that what are the name of those enzymes? There are a variety of enzymes that are actually allosteric enzyme. I just I wrote two examples. One is the phosphofructokinase. This enzyme belongs to glycolysis. Glycolysis, you know very well, it is the metabolic pathway of carbohydrate metabolism, glucose metabolism. And second enzyme is isocitrate dehydrogenase. This belongs to Krebs cycle. Krebs cycle is also a pathway of carbohydrate metabolism. Phosphofructokinase has its activator as well as inhibitor. Similarly, isocitrate dehydrogenase has its activator as well as inhibitor. What are the inhibitor of this enzyme? The inhibitor of this enzyme is actually ATP. In this case, the inhibitor is ATP. In both cases, the inhibitor is ATP, but the activator is, in this case, for phosphofructokinase, the activator is ADP. Means when ADP is there, definitely it means ATP is absent. So when ADP comes, it will activate the enzyme, it will order the enzyme to start synthesizing ATP because ATP is less. So that's why. And for isocitrate, the activator is what? Adenosine diphosphate and nicotinamide adenine dinucleotide. NAD and ADP, these are the activator. So other enzyme that we have also have this kind of activator and inhibitor. You can just memorize these two examples for your exam. I hope you got this lecture. See you in the next lecture. Take care. Bye bye.